I'd like to welcome up Mr. Brian Mitchell from Ph.D. from Satellos or Satellos? Satellos.
Satellos.
It's a biotech company focused on muscle regeneration. Dr. Mitchell is a senior vice president of medical and science affairs and has 15 years of combined scientific and biotech experience from stem cell research to business and corporate development. So welcome, Dr. Mitchell. Let me get you figured out here. Does it tell us?
And if you're in Canada or the US.
Oh, yes.
Satellos here, Satellos.
Oh, there you go. Okay, it's both right then.
Go either way.
Right.
So thank you, everybody, for being here. Of course, thank you to the organizers, and a special thanks to Neil. So we are going to switch gears a little bit and talk about Satellos or Satellos, however you prefer. I just want to put some expectations forward first. So we are not currently in the clinic for FSHD. So we are a Duchenne focused company at this time. But thanks to Neil and some support by FSHD Canada, we have started to seriously think about FSHD. But we're a very small, relatively new company. And so we are trying our best to get our first step right. And if this technology does have promise in Duchenne, we strongly feel that it could have promise in FSHD. So my real goal for today is to socialize with you in this community, get you thinking about what it is that we're doing so that hopefully you can track us and hopefully in the fullness of time, we can see a trial outside of Duchenne and other muscular dystrophies and most certainly in FSHD. So get your cameras ready, most exciting slide. These are my disclaimers. So we are a publicly traded company here in Canada. I won't spend much time here except to say that we are developing a drug, SAT-3247. We believe that this could be a highly, what we call in biotech, a disruptive drug. That's a fancy way of saying a very different way of thinking for treating degenerative muscle diseases. A very different approach. Lots of times we think about drugs in this space as maybe ways to slow down degenerative disease, which would be fantastic. That's a great goal to have. When we think about muscle regeneration, if everything works out as it sometimes does in experimental models, we could improve the muscle from where it was when you started. So that's what we call a blue sky opportunity. We hope that that's what comes about, but you never know until you try. So that's what we're trying to do right now. 3247 is currently in the clinic. We've treated healthy volunteers. We are currently treating adults with Duchenne in early safety studies. And in 2025, we hope to launch a phase 2 program, our first sort of next step of the trial process where you actually try to show whether the drug is having an impact on disease. So I'll tell you a little bit about Duchenne and our story in Duchenne. I promise you I will link it back to FSHD, but it's important for us to get our story across from where we started, and then hopefully build some bridges to FSHD. So our mission in Duchenne is to try and correct what we believe is impaired muscle regeneration. So you may not even be familiar with the concept of regeneration, maybe you are, but muscle is one of the very few systems in our body that when operating normally has the capacity to repair and regenerate. It's not science fiction, that is an absolute truth, to the point where lots of nerdy scientists like myself have used muscle tissue as a model to study regeneration because it does such a good job. A lot of the science is homegrown here in Canada. We have a lot of researchers focusing on muscle regeneration in Canada. Our scientific founder, Dr. Michael Rudnicki, was part of an early team who helped describe the cells in the muscle that are actually responsible for this capacity. And the science of Satellos is based on those findings. And so when we think about Duchenne, we think about this as a disease. Of course, it's genetic in nature. We know there's a mutation that maps to a particular protein. When individuals don't have that protein, the muscle tissue becomes very fragile. It breaks down. This is not contested by Satellos. What Michael discovered is that this gene has another role inside of muscle stem cells that helps them do their job, which is to respond to damage and build new muscle tissue or repair that damaged tissue. So in Duchenne, we believe there's really a two-hit problem. You have muscles that are fragile and break down from regular daily activity. These young children walk around, they stand, they play in the early years of their life, and that causes serious traumatic damage to their muscle. The muscles try to repair themselves. You can see that when you physically look at the muscle tissue, but they clearly don't do a good enough job. And year over year over year, muscle tissue is lost in these individuals. It's not all at once. It does take years. And that's because you have a repair system that tries to address this problem. So Satellos, in a nutshell, is trying to fix that aspect of the disease. How can we take this issue with regeneration and give it a little boost, help it out, so that the tissue has a better shot of keeping up with the damage? So that is our goal. When you look at the landscape of Duchenne for different treatments, there's a great deal of focus on the genetic issue. We've got a gene, we've got mutations. Can we fix the gene? Can we skip over the mutation? Can we provide a miniature version of the gene and try to express the protein that should be there that's not in there in these patients? And will that provide some additional strength to that muscle tissue and keep it from breaking down? All of these genetic approaches are fantastic. They all have a great mission in mind. Satellos is focused on something very different. We know there's a mutation in the gene, but we have this other function that we're trying to address. We look at the stem cell, we look at regeneration. That, in the fullness of time, could also be corrected by genetic means. But to make a long story short, that's very difficult to do with stem cells. They're far and few between, and to correct them may be a bigger challenge than correcting the muscle at large. That being said, we have figured out where this problem is in the stem cells. We sort of know the issues with regeneration, and we have developed a drug that helps to correct that problem. So we are giving the tissue a chance to try and keep up with the damage and stick around for longer. And we're doing that with a pill. This is a once-a-day oral medication, a classical sort of drug development paradigm. We have a protein that we're trying to interact with, and so you build a little piece of chemistry to change a decision inside of these cells and have them go through that process of regeneration more sort of robust than they would have otherwise. So what does this look like? So we'll get into some data, and I will try not to bore you with details. We've done lots of studies in mice. We'll talk about some mouse studies that we've done with Neil, and we've moved into larger animal models. It's really important when you're assessing these kinds of things to take into context that these are preclinical animal models, as we call them. These are experiments. These are not people and patients, but the ultimate goal here is see if your idea is working experimentally, see if it's safe, and then we bring it to patients to see if it can have real impact. So what I'm showing you here today is some data from a Duchenne dog model. Dogs actually get Duchenne muscular dystrophy, so these are not artificially produced animal models per se. Colonies are carried through year over year, but dogs get Duchenne. This was a 4-month study where we took dogs that were about 9 months of age, and I tell you that because in sort of dog Duchenne terms, this means the disease has really come on in these animals. They're exhibiting signs of severe weakness. Often drug trials will take place in younger dogs when they're still growing so that disease doesn't get so serious. Because we were trying to improve the muscle, we didn't see this as too much of an issue. But we treated these dogs for 4 months with our molecule. We assessed a whole whack of things. We looked at their muscle tissue before and after, but most importantly to Satellos, because we're focused on repairing things and regenerating them, we should be making them better than what they were. We look at function. And so we looked at the strength of these animals. I need a pointer. You still hear me without the mic? Is that fine? So we have strength on the x-axis, age on the y. You see these black trace here, gray trace there. These are Duchenne dogs in gray. Black line is healthy dogs. Hopefully what you can appreciate is that Duchenne dogs are always weaker than healthy dogs over time. This is because their muscles physically deteriorating the animals present that in a loss of strength. The teal line and the purple line are the two animals from the Satellos study. Hopefully what you can appreciate there is that those two dogs were in fact sick. They started off weaker than typical Duchenne dogs. And after 4 months of being on this drug, their strength was effectively brought back to normal. Now, this was the best metric we saw. We did look at other muscle groups. We looked at flexing and extending. They did not all get back to normal, but we saw an improvement in everything that we did. What I don't have for you here today as well is a whole whack of pictures that show very nasty looking muscle from biopsy at the start of the study, which is what you would expect in Duchenne, looking like very healthy muscle at the end. All of this is available on our website if you'd like to take a look. But the take home here is that if you give the muscle back its normal, innate capacity to heal itself, it can and it will. That's the goal of the drug. And just to remind you, this is an experiment in an animal model. We have to see if this translates to patients, but we think that this gives us a reason to try. So this is Duchenne. This is very similar to what we've seen in mice. And with the help of Neil, we then tried to look at different diseases. We've looked at a number, and FSHD is one of them. So this was a study that we did in a mouse model of FSHD. Again, 3 months of treatment with the drug. And to kind of orient you in the same way, this black line on the top is healthy mice. The gray line on the bottom are FSHD mice, and this purple line kind of in the middle there are FSHD mice with the drug for 3 months. And so again here, the pattern is that these FSHD mice are improving. Now, what you can't really see from this data is the first comparison. So ultimately, we didn't start off, earlier in the weeks there, but there's a separation immediately after 4 weeks. So if we had have tested these mice for strength at the beginning, you can see that the drug-treated mice, pre-drug, and the drug or the no-drug mice all started off the same, and they're separated. And the separation is an improvement in strength. So often when you look at trials for FSHD or for Duchenne, we look at placebo-controlled trials where the goal is to try and slow the progression from the treatment of the drug. Here we're seeing an improvement in the muscle strength, which makes sense if you're bringing the muscle to a better place. So these sort of animal model experiments are what encourage us now to try and think about going into patients. We're doing that in Duchenne. And of course, in the fullness of time, we hope to do that in other indications, should the science lend itself. So very quickly, I will wrap up by saying that we have treated healthy volunteers. We have an excellent safety profile for this drug, safe and well tolerated from the initial study that we've carried through. We hope to release similar data on this in the adult Duchenne population later in this quarter. We'll keep you updated through our website. It will be always a press release affiliated with these things, but the drug seems to be behaving very, very well in healthy volunteers. We've seen initial data that tells us the drug is being handled in patients in the same way that it's being handled in animals with respect to drug levels over time. So there's a whole process of science called pharmacokinetics, which is very, very important when you first transition from animals into patients. You want your drug to be handled in the same way and to hang out in the bloodstream for the right amount of time. So we've been able to confirm all of that in healthy volunteers, which is great. And we're hoping to do that in patients. I'll show you some timelines here, but it's always best just to stick to the website as things often change. But as I said, we're in Duchenne patients now, not attempting to demonstrate what we call efficacy, but mostly trying to ensure that that safety profile is maintained and the drug is handled in these adult patients in the same way that it's handled in healthy volunteers. And then later on this year, we hope to transition to a study that truly tries to look at whether this drug is having an impact on Duchenne in patients in the same way that we are seeing in these animal models. And really, with that sort of early data in hand, I think would drive confidence that we're seeing translation of this mechanism from animals into people. And that's where now I'll transition over to FSHD.
Make some promises, will you?
Yeah.
This is a crazy promise.
So really what we believe this drug is doing is it's helping muscle tissue respond to damage, okay? So in Duchenne, obviously we have a very different genetic problem from FSHD, which, is a very different problem from many other genetic dystrophies. But the unifying feature that we think is present is muscle damage. So SAT 3247 is not designed to fix a Duchenne issue. It's designed to help muscle stem cells respond to damage and produce new muscle tissue. That is all it's doing. And if there is no damage, this drug does absolutely nothing. It needs damage to be there. It needs these cells to be responding and trying to fix the muscle tissue. If that process is occurring, whether this is in Duchenne animal models, FSHD animal models, or even completely healthy mice where the muscle has been deliberately damaged, every single time we have looked at this drug on board, it improves the regeneration process. So that is the theoretical prospect for FSHD. We know that there is muscle damage in FSHD, and there's a chance that a molecule like this could help the muscle tissue to repair itself. That's what we believe we're seeing in the animal models across the board, and hopefully in the fullness of time, something that we could see in patients. So I wish I could tell you more today about what we're going to do in FSHD and when. I know that that is the only thing that matters to you. Please just keep emailing Neil. He calls me every day. He will absolutely let you know. But we're absolutely excited about the idea of doing something in FSHD as a company. We know that this would be incredibly meaningful to you. And selfishly, as a company, we think that this is a great place for us to be because the indication fits the bill for how this drug works. And so in the fullness of time, hopefully on the success of some work in Duchenne, we can move into FSHD and to be honest, other dystrophies and indications where there is muscle damage. So if I have confused anybody, please ask questions. I do have to leave shortly after this, but you can always go to our website. We've got lots of forms you can fill out there and contact information. So we're an open book, and thank you for your attention.
Okay, the agenda actually has time for one question, and then I'm gonna have Lawrence and Amanda come back up because I totally spaced out taking your picture. So we have to take a picture before we can go on. Question over here, loudly.
Very quickly, it might seem like an odd question, but the muscle has to exist. Is it muscle wasting? If the muscle is not there, obviously you wouldn't repair something that's not there.
That's right. That's right. So you need to have the muscle in order to make muscle. And so, you know, there's different ways of thinking about wasting. So if you think about atrophy versus muscle damage, so this drug does not impact the sort of atrophy, hypertrophy system. Really where it plays is if you have a severe enough injury to the muscle fiber that would otherwise require sort of stem cell mediated repair, which is how we repair muscle. It's how everybody repairs muscle. This drug can enhance that process, or it's believed to enhance that process. Preclinically, we can do confident. Clinically, we need to find out. But if there is no muscle left in an extreme situation, it is not muscle in a pill. This will not create muscle from nothing. It can enhance an ongoing repair process. It also will not initiate the repair process. So damage and injury causes a whole whack of growth factors and signaling to occur. That kicks the whole process off. That tells stem cells to start dividing. This drug influences how they divide and gives them a bit of a nudge to go down the pathway of making new muscle tissue. So a few things need to be happening in order for the drug to happen, but in FSHD, if you still have muscle left, and if that muscle is still incurring injury, which we believe from looking at muscle in FSHD, there's lots of evidence to suggest that injury is occurring and death is occurring to those cells. That triggers everything that we need in order for this drug to work. And we see that preclinically. Of course, we remain to see that clinically.
Thanks for that.
Thank you, Ryan. Thank you.
Thanks, Brian.